Alright, Chapter 9, Part 3. In Part 2 we spoke about glycolysis, which is the first of the three stages of cellular respiration. In this one we're going to be looking at the citric acid cycle. Now the citric acid cycle, part of cellular respiration, will only occur if oxygen is present. If oxygen is not present, the pyruvates that we form during glycolysis will go into fermentation. So this will only happen if oxygen is present. In the last part of glycolysis, we made the pyruvates. We're going to see those turned pretty quickly into acetyl-CoA, which links the cycle to glycolysis. So let's check that out. So pyruvate right here, it is short-lived. And we can kind of see that on the image here. Right here it's pyruvate, and right here now it's acetyl-CoA. So the carboxyl group, highlighted in gray here, the COO group, it's removed from carbon dioxide, and it's removed to form carbon dioxide. After the carbon is removed from here, all that's left is a 2-carbon sugar. This 2-carbon sugar at this point, right in this portion, now in the mitochondria, is oxidized, and we form some NADH, an electron carrier. This is followed by the attachment of a sulfate compound, a sulfur compound, forming acetyl-CoA. We have coenzyme here, and now we have our sulfur compound, so it is now acetyl-CoA. Acetyl-CoA is now ready to enter the citric acid cycle, or as you may know, it's the Krebs cycle. Here we can kind of see an overview snapshot of the citric acid cycle. We had our 3-carbon pyruvate come in. Carbon is removed as carbon dioxide, leaving a 2-carbon sugar. It's oxidized. We have an electron carrier now. Acetyl-CoA comes in along with the sulfur group, and now we have a new product, a 2-carbon acetyl-CoA group, which will then enter the citric acid cycle. This cycle takes place in the mitochondrial matrix. The cycle oxidizes organic fuel derived from this pyruvate that we had coming in, generating 1 molecule of ATP, 3 molecules of NADH, or electron carriers, and 1 molecule of FADH, another electron carrier, per turn of the cycle here. So we're going to be looking at this in further detail. So the citric acid cycle has 8 steps. Each is catalyzed by an enzyme. We can see acetyl-CoA coming in and linking up with oxaloacetate, which is then transformed into citrate here. It will work its way around and eventually become oxaloacetate once again, which makes this a cycle. The electron carriers, NADH and FADH, produced during this cycle, as we'll see shortly here, relay electrons extracted from food to the electron transport chain, which is linked up with more or less step 3, oxidative phosphorylation in cellular respiration. So once again, another enzyme acting on the citrate here, removing some water, becoming isocitrate. So we've changed our molecule a little bit. An electron is picked up by NAD, becoming NADH, a little bit of carbon dioxide released, and now we have alpha-ketoglutarate. More removal of carbon, and another electron removed, and we have changed our molecule once again. Our molecule is then phosphorylated by the addition of this phosphate group, which is immediately used to phosphorylate ADP, creating that 1 molecule of ATP that we spoke about previously. We now have a slightly different molecule, which then loses 1 electron to the electron carrier FAD, becoming FADH. Now have a slightly different molecule. Add in a little bit of water to this molecule, changing it once again. And with the removal of another electron to the electron carrier NADH, we are back at the oxaloacetate state. Once again, to recap that screen that we saw once at the beginning, we have NADH, NADH, and NADH. So we have 3 NADHs formed. We have 1 molecule of ATP and 1 FADH formed as well. So 4 electrons are picked up, 1 ATP is produced. You're probably starting to freak out at this point. Don't make a huge deal out of this. What you need to know is acetyl-CoA goes into it and links up with oxaloacetate. This really is the one word that you'll want to remember. These other ones aren't nearly as important. You are welcome to memorize them if you'd like. But what we need to be able to do is remember acetyl-CoA, oxaloacetate, and be able to understand how these electron carriers are picking up electrons, as well as where the carbons are going as they change. So we see 2 carbon dioxides coming out. We'll need to kind of keep in mind the products that are made from this cycle. So products, what's being made, some electron carriers, some carbon dioxides, some ATP, and that's kind of what you're going to need to take away from this. So we've talked a good amount about the electron carriers picking up the hydrogen electrons. Those are going to be going to the electron transport chain, which will be used to ultimately drive oxidative phosphorylation. And oxidative phosphorylation is definitely where we pick up our ATP. So now looking at the electron transport chain, we can see the pathway right here. It's a slow downward process that's very important. It's occurring in the cristae of the mitochondria, and the electron transport chain itself generates absolutely no ATP. We see no ATP coming out. Most of the chain's components here are proteins. Protein, protein, protein. Looks like they're labeled in Roman numerals here, 1, 2, 3, and 4. These exist in multi-protein complexes. So there's multi-proteins forming each one of these complexes. The carriers themselves, right here, are reduced and oxidized as they accept and donate electrons. So they'll accept an electron, donate an electron, accept an electron, and then donate it. Now that's donated, it accepts it and passes it down once again. We can see the electrons being donated by NADH and FADH above. Take note between these 2 electron carriers that NADH is dropping its electron off in the first protein complex, whereas FADH is dropping it off in the second protein complex. This does have a slight difference in that this electron donated by NADH will go through more proteins than FADH will. The FADH got dropped off later. FADH's electron will not go through this first protein, which makes this electron a little bit more able to produce more hydrogen ions, as you'll see as we continue to go through this a little bit more. So moving into the electron transport chain, this is in the cristae, yes, but specifically remember those cristae are the folds in the inner membrane. So this is actually in the inner membrane within the cristae of the mitochondria. And with each protein that the electrons pass through as they move down the electron transport chain, hydrogen ions are pumped from inside the matrix to the intermembrane space, which is the space between the outer membrane and inner membrane of the mitochondria. This creates a gradient, a hydrogen ion gradient. This will come up in the Bozeman video that I have you guys watch a little bit later after we get through this discussion. The hydrogen ion gradient that's now created will be used to drive the formation of ATP in the second part that follows the electron transport chain, the actual generation of ATP. We'll see that oxidative phosphorylation a little bit more on the next slide here. So as you can see, there's a series of compounds in place to carry the electrons. We spoke about those electron carriers. These are called cytochromes. So there's quite a few cytochromes, as we can see. As they're passed down, the electrons, they drop in free energy as they're moved, ultimately towards this oxygen down here, forming water. Again, the idea of this process is to release energy from those electrons in manageable amounts so that we can force the hydrogen ions up to the inner membrane space, which will inevitably drive the process of ATP formation. This is also in the inner membrane of the mitochondria. We can see the inner membrane space up here and the matrix down here. These are the hydrogen ions that are now existing out in this space as a result of this process. These electrons move down, they put the hydrogen ions out into the inner membrane space. So what's the significance of these hydrogen ions? Well, that's what we're going to check out right now with the function of ATP synthase, which is this complex right here, including the stator and the rotor. As you can see, it's embedded in the membrane of the mitochondria, specifically the inner membrane. And this image illustrates the hydrogen ion concentration gradient that once again was created during that electron transport chain. Hydrogen ions then move back across the membrane, passing through channels right here in the ATP synthase. ATP synthase uses the exergonic flow of these hydrogen ions to drive the phosphorylation or addition of phosphates to ADP, and this will form ATP. This interaction that we see going on within ATP synthase is an example of chemiosmosis, or the use of energy in a hydrogen ion gradient to drive cellular work. It works first by having a hydrogen ion enter a small channel in the stator, which is then connected to the rotor. The rotor accepts the hydrogen ions, and it causes the rotor to rotate, as we can see by the arrow there. Each ion will rotate one time around the rotor before exiting through another little channel, and it's now inside the mitochondrial matrix. So as this top part of the rotor is rotating around as a result of the energy from the hydrogen ions, the bottom part is rotating as well. And the bottom part of the rotor contains catalytic enzymes that allow ADP to be phosphorylated, becoming ATP. Now I mentioned earlier that the electron transport chain does not generate ATP, which is true, but we see that going on right here. This process on the screen is not part of the electron transport chain, but it does make some ATP, and it relies upon the electron transport chain in order for that to happen. So this is really immediately after the electron transport chain. And it's the electron transport chain that creates this hydrogen ion concentration gradient necessary for this to work. This is known as a proton motive force. So to put the whole oxidative phosphorylation thing together, we see the electron transport chain and chemiosmosis kind of working together here. Once again, as the electron carriers dump off their electrons to those cytochromes, the cytochromes carry them across protein complexes. And each time an electron goes through a protein complex, we see hydrogen ions pumped from inside the mitochondrial matrix to the membrane space. And that creates the concentration gradient necessary for the hydrogen ions to go back down through the ATP synthase to crank out the ATP that we see going on here. This process of the chemiosmosis will produce a total of 26 to 28 ATP. And recall that earlier productions of ATP were much lower, 2 ATP. During respiration, the energy flows in this sequence. It goes from glucose to NADH to electron transport chain to the proton motive force to ATP, producing a total of 2 plus 2 plus 26 to 28, equaling 30 to 32 ATP.